As the background of the study, Ecomia crassipes or water hyacinth is a worldwide recognized aquatic weed. This species originates from Amazonia and spread throughout the tropical, subtropical, and other warm climate regions throughout the world. Being a widely distributed weed species, it poses major problems in the different freshwater bodies, earning its name as the world's worst aquatic species. Since 1990s, this water hyacinth has posed serious challenges. In 1998, due to lack of natural enemies, the plant spread and attains its peak infestation about 17,200 per hectares on the Kenyan side. Several management options were considered to control the weed, including physical, biological, mechanical, and chemical methods. In the Philippines, water hyacinth has been considered as one of the most unwanted waterborne plants. This species chokes water intake points and rivers and dominate the aquatic vegetation cover in ponds and lakes. Oftentimes, the presence of water hyacinth in rivers and lakes causes flood because they blocked the outlets of these water bodies. In Lake Lanao, water hyacinth becomes unwanted due to its serious impacts on the water dams and lentic water bodies, which are the major source of water for hydroelectric plants along Agos River. Thus, the aim of this study is to show water hyacinth biology, chemical composition, and its negative impacts on the aquatic ecosystem, biodiversity, economy, and human well-being, as water hyacinth is challenging the ecological stability of freshwater bodies. This also identifies the insects living in this habitat and explaining the fact of how can they suppress this invasive species. Moreover, this research proposed ways in how to manage and control this water hyacinth and how do insects suppress the invasion of water hyacinth, particularly in an aquatic ecosystem. The RRL also explained the management of water hyacinth. Particularly, it involves mechanical control, chemical control, and biological control, where insects are the main agents to attack water hyacinth. And as the methodology of the study, there are two factors that are being explained: the factors that accelerate success and the factors that limit control. And particularly, the researchers focus only the factors that might accelerate control. And so, the method of this proposal only focuses on the factors that might accelerate control, which basically involves biological agents or the biological control of suppressing water hyacinth, specifically insects. The study will be conducted at Lake Nangkabaloy, Lanao del Norte, where water hyacinth is abundantly present. Lake Nangka was selected based on its availability and accessibility. This study will be carried out in a complete randomized design or CRD with 20 sampling plots or 1 by 1 meter. Sampling plots will be selected randomly along the lake shores by drawing lots in the box without looking at it. The 20 sampling plots were numbered according to its number that was drawn. And for the sampling procedure, 100 meters plastic straw will be used to locate the sampling plots, which was set at 5 meters parallel distance. A wooden frame 1 by 1 meter will be used to delineate the sampling plots on the ground 26. For data collection of insect samples will be done from 9 a.m. to 2 p.m. with the idea that during these hours, the insects are actively doing activities. Without the sampling plot, all insects found within the above water component will be collected, be counted, photographed, and preserved. Insects will be collected using insect nets and tweezers. The insects will then be preserved in a solution of 70% isopropyl alcohol prior to actual identification in the laboratory.